In this video, we will discuss about some of the challenges and optimization of the CAR construct in NK cells. Last time we have discussed about the T cells or the CAR T cells challenges. In this video, we will discuss about the challenges of the CAR construct in the NK cells. So, and most of these challenges or problems are similar to those we have seen in the T cells. First one is this NK trafficking. That is, the challenges is to improve NK cells migration and infiltrations to the tumors, because in order to reach to the tumors they have to go through the tumor microenvironment, which creates a physical, not only just the physical barrier, but it will suppress the immune response, as it will have the immunosuppression environment. And these things can be overcome by genetically engineered these NK cells to express these silence these chemokine receptors that can take care or neutralize the immune suppressions that is present within the tumor microenvironment. Or the other things that one can do is intratumor delivery, is directly deliver to the site of tumors, or enhance the gradient, chemokine gradients towards the tumors. And the next thing is this tumor microenvironment. It will reduce the, as we have discussed, it will reduce the NK sensitivity to immunosuppressive signals. That is, the immunosuppressive signals are present in the tumor microenvironment. You need to disrupt these immunosuppressive signals, and some of those signals important. Some of the main signals are this TJ beta signaling, adenosine signalings, or checkpoint signalings. TJ beta signalings promotes the tumor cells to have these proliferations and migration properties, and this adenosine signalings helps the tumor microenvironment to maintain its pH that results in the immunosuppression effect. This checkpoint signalings interacts with the ligands such as PD-1 ligand that are present on the tumors, or PD-1 receptors that are present on the tumors, and PD-1 ligand that are present, PD-1 receptors that are present on the these immune cells, or in this case NK cells, and PD-1 ligand that are present on the tumors. So those interactions might result in the suppressions of the immune activations. Next one is this NK persistency. The lifetime of the NK cells is also important, as it is difficult to traffic the NK cells to the site of tumors. They need to have a prolonged life so that the efficiencies will also increase. So in order to do that, the systematic cytokine administrations, as the NK cells are sensitive towards this IL-2 ligands, so IL-2 continue the administrations of such cytokines. And here they have mentioned about the IL-15 expressing NK cells. That is, NK cells are able to express the cytokines that is required for their own survivals. Third one is this cytokine induced memory like NK cells. It is similar to the second step, whereas you can consider this as the second one is the one of the example of the third one, that is cytokine induced memory like NK cells. NK cells are able to produce cytokines that is necessary for its own survival. And the third one is this CAR construct. Right now the CAR construct are mainly optimized for the T cells. They need to be optimized for the NK cells also, such as the domains needs to be switched, cellular domains needs to be switched to this NK GD2 in the CAR, and whereas in the T cells they use different receptors. The transmembrane domains can also be replaced to DNAM to beta 4 NKGT2, and intracellular co-stimulatory domain they can be replaced by DEP10 DEP12, as they are more related to the NK cells, where compared to the CD3 zeta, which is related to the T cells more. Third one is this NK source. One need to have a large number of NK cells so that they will have a more efficient effect and to large populations of the tumors. So one of the ways to do this thing, and also to reduce the heterogeneity and the manufacturing complexity, this also needs to be there, so that it will have a similar effect on all the tumors that it encounters within the host. So these things can be done one by using this NK92 cell lines, or taking the blood from the patient, such as peripheral blood, umbilical cord blood, or hematopoietic stem cells or induced pluripotent stem cells. This source can be used to extract NK92 cells. Third one is this NK anti-cancer activities. That is to increase the or enhance the CAR-NK cells functions. These things can be done by mutating some of the genes that leads to the suppressions of the NK cells, such as the CAH-SH gene or CALMH2 gene, or by overexpressing this DNAM1 or NKGT21. These things will also helps in the NK cells survival. And now we will quickly discuss how one can optimize this CAR construct towards the NK cells. As cellular domains, it should be specific to the tumors against which this CAR construct is developing. So this can be customized based on tumors to tumor. The hinge domain in the case of this T cells, CD8, CD28, and Ig1 and Ig4 are commonly used, whereas in the case of NK cells, NK cells can also use one of these domain. The changes lies in the transmembrane domain. In the case of this T cells, these domains can be used CD3 zeta, CD8, CD4, CD28, and whereas in the case of this NK cells, this DNAM1 2B4 NKGT2 transmembrane region can also be used. And for the co-stimulatory domain, CD28, 411B are preferred for the T cells, whereas in the case of NK cells, CD16 is preferred. And similarly, the other co-stimulate or activations domain to activate the NK cells, this DAP10 and DAP12 is this CD3 zeta is preferred for the T cells, whereas DAP10 and DAP12 is preferred for the NK92 cells. So thereafter we can be able to, if we can switch these receptors to the NK92 compatible receptors, we will be able to make a CAR construct that is optimized for the NK92 cells. So I hope you understood how these NK92 cells are able to can be used for the cancer treatment by optimizing the CAR and by also preparing strategies to overcome the challenges for the NK cells to be used for the diagnosis, especially in the solid tumors. If you need to understand more about the challenges and optimizations of the CAR construct in NK cells, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.